Good afternoon, everybody. This is Laura Tew, host of today's Florida Sea Grant Bite Size Science. And we're in for a special treat today. We have Dr. Dale Laughinghouse from the Fort Lauderdale Research and Education Center and Dr. Vincent Encomio presenting today on cyanobacteria, public health, and potential impacts on your pets. As always, your cameras and microphones are muted, but please feel free to put any questions in the chat box. So let's go ahead and get started. Dr. Laughinghouse, please take it away.
Thank you. So let me get this thing started now. Okay, hello everyone. Thank you for coming. I'm going to start talking today, and then afterwards Dr. Comey will talk, and I will begin the conversation about cyanobacteria and public health, and then we'll go to some crop health as well. So the outline, I'm going to give a review a bit of what we talked about last week. I don't know who of you guys were here last week when I talked, so I will talk a bit about cyano, and then about blooms and proliferations, and then the new material will be about cyanotoxins in public health and plant crop health, and then Dr. Comey will talk about microcystins and dogs and pets. So again, you guys saw this last week, but just going again, what are cyanobacteria? So cyanobacteria are photosynthetic bacteria. They're the plastids in the plants or other eukaryotes. They have various features for survival, and those are some gas vesicles or aerotopes that can help them with the scumming, so it helps them go up and down the water column. Some of the species can fix nitrogen. They're about 3.5 billion years old. And why is that important? Because they've outlived a lot of things, right? But to just think about that, some of these gene clusters, some of these toxins we'll talk about a bit later today, they evolved 3.5 billion years ago. So I get a question a lot of times, oh, is it safe to eat this? Is it safe to eat that? Well, microcystin, which is one of the compounds we'll talk about, that evolved at the beginning of evolution of cyano. So theoretically, anything that evolved from that should have the cluster, right? Some cyanobacteria have lost it over time, but in theory, all cyanobacteria are potentially able to produce, let's say, microcystin or a toxin as well, other ones. They can use both oxygen and hydrogen sulfide as, for energies as electron donors. They're found in many habitats and they're morphologically diverse. Like I showed you guys last week. Here are different habitats are found. You know, they're found on animals, on structures, on the soil, you know, on the benthic area and on wet rocks, and here, as you know, in the plankton. And so that's where they're found. But what is a bloom? Again, this is material that we looked at last week. Just to remind you guys, so a bloom is usually a dense growth of algae. They can produce toxins right here. They're able to also harbor pathogenic bacteria on their mucilage, and they can contain taste and odor compounds. This is something very interesting, and we're not talking about it today. Sometimes I get the question, okay, it tastes funny, or does the taste and odor compound, does it mean it's toxic? Not directly. There is some correlation that geosmin and the microcystin production may be correlated, but it's not totally correlated. And also, we talked last week about treatment, and the way you treat taste and odor compounds are different than the way you treat algae or you treat the toxins themselves. Again, so blooms, some pictures of blooms. You guys saw this last week. Here's just different ways they look like in the water. You know, these are pelagic blooms, and we always hear about planktonic blooms or pelagic blooms. But, and here are some of the species you see, right? They're very, very different. But blooms can also be found in other areas, like I showed you last week. You see this kind of stuff you guys see in the canals that usually around, you know, in this kind of, we call it the benthic mats. So you see benthic cyanobacteria. And if you look at this here, this came out last week, you see that we have different, in this paper, we talk about different toxins. So in a second, I'll talk about the different names, but anatoxin and saxitoxin, these are neurotoxins. Cylindrospermopsin and microcystin and algilarins are hepatotoxins. Neurotoxins on this side, they affect your brain, right? And the cylindrospermopsins and microcystins and nodularins are hepatotoxins, the liver, right? So what I'm trying to say here is that you know, when you look at the scum, you see, okay, oh, it's scum forming, it's producing toxins. Maybe it is a microcystin or whatever. But we also have the benthic cyanobacteria, which are these looking ones, that they produce compounds. And those compounds are actually different than the scum formers. And a lot of times I think, and I've talked to colleagues, that a lot of these deaths of pets that you see, I know Vince is talking about this later, they are probably produced by the benthic cyanobacteria and by these neurotoxins because they kill very quickly. But the problem is that they degrade very quickly, and I'll show you that in a second, which means that when you have the sample and send it out for analysis, by the time you get it back, by the time it's analyzed, you won't see the the neurotoxin in there anymore. Again, just one more slide from last week. Toxic cyanobacteria are also found in mats, soil crust, and benthic marine mats, okay? So again, remember, this is the reason I told you guys they evolved 3.5 billion years ago. Toxin production started back then, not everything, but some of the gene clusters. And they're produced in all organisms now, or can be produced, they're putatively produced, okay? So now going back to the new material for today. So now you guys remember now everything from last week. So new material. Toxic cyanobacterial blooms. They're not new. It's nothing that started now. I think some people have heard me talk before, and I comment about the Red Sea of Moses turning it red, and you know the Bible going back talking about how you know blooms back then. But so in 1188, this is you know people had started noticing water changing color. You know, bright green, which would be microcystis, and scarlet would be a planktothic strobescens. So these are two different cyanobacteria. Remember, microcystis is the cyanobacterium, the cell of the species, and then microcystin is a toxin. You guys will see that later. But also, and then 1833, livestock deaths were starting to be noticed, and they started correlating livestock deaths with this ugly water, colorful water, or I think it's beautiful water actually, it's job security. Then in the 1870s, these were starting to be recognized as cyanobacteria, and that was a lot of taxonomist studying. So taxonomy is a study of the organisms, of the of naming species. So they would go out and collect and name species. And in the 1980s, toxins started being studied. We're in 2020 now. So we're in 2020, and it's not new, and it's been going on for a long time, but we still don't know a lot. So the poisonings or intoxications have been identified or been documented in many metazoans or many animals and insects, like honeybees over here, and in fish, and in people, and in crocodiles, and in a lot of different things. Just here's to show you guys that it's not just that these toxins can affect many different organisms and also plants and organisms that are not just humans or pets, okay? So we're talking about toxins and toxins and toxins, but when did it start? I mean, I told you guys that it started that, you know, the 1833 and the 1980s, but when did, let's say, the WHO get involved or the EPA start thinking about this? It's in 1996 about, so this is from something happened in Brazil, in Caruaru, which is a city here, that around 100 patients developed acute liver failure and 76 died. And that was the wake-up call of for cyanobacterial toxins, or in this case. So what happened was that there was a drought. So those that went to my talk last week, I showed something about scum forming in water when there was water was stagnant. If you guys remember that the cell concentration became a scum. So in this case, what happened here was that there was a drought, and the dialysis center was run by the municipal government. So they said, go grab your water truck, go out to the reservoir and grab water because it was a drought. So the intake pipe for the reservoir, for the water for the city was above the level of the water. That makes sense. So what happened? They went out there, they grabbed water from the reservoir, they put it in a water truck, and the water was green scum. Okay, what did they do? Oh, they wanted to clean it. They put chlorine in it. What does chlorine do? It lyses the cell. So it let all the compounds, the microcystins in that case, go out of the cell into the water. And that water was taken to the dialysis center and pumped into everybody. So that's the story behind that. And it killed people. So of course, that's when the WHO got involved and other countries started getting involved. So yeah, now you guys know the story, right? So now we're gonna go into cyanotoxins. I've talked about these names, but let's go into some of what they are, okay? So the groups of cyanobacterial toxins. There's more groups, but we can classify in these different ones. So you have the neurotoxins, okay? And these are some of the names: anatoxin, saxitoxin, guanotoxin. But most are not cell bound, okay? And why is that important? It is important in management because these that are cell bound are the ones that when you kill the cell or lyse the cell, they go into the water column, okay? But anyway, in general, the neurotoxins, they cause death by respiratory failure. The liver toxins, the hepatotoxins, they're cell bound, and they affect, they do death and illness by liver necrosis, cancers, tumors, liver failure. And these are most cell bound. And the problem here is chronic exposure. So small concentrations over time and not so much acute exposure. So other ones you have are dermatotoxins and endotoxins and other peptides. It's a lot. There are thousands of compounds that exist. So just for you guys to have an idea what they look like, microcystin, there's about a bit over 250 variants. And then there's nodularian. These are two that are found in Lake Okeechobee, for example. What I don't need you guys to remember the structure. The only thing I want you guys to know is big, and that the half-life can be days to up to 10 weeks. So the hepatotoxins, the microcystins, they stay in the water for a long time. Of course, that's dependent on water chemistry, temperature, and light. And remember, these are also in the cell. So what happens at the end of a bloom, the cells lyse, or if you treat it, they lyse, the compound goes out, okay? And they stay in the water for a long time. A little bit more about microcystins. How do they act? They inhibit protein phosphatase, okay? And this is what causes these cytotoxic and tumorigenic effects. Again, like I said, chronic exposure is a problem, which causes tumor development due to mutagenicity. They're responsible for the death of animals, including humans. It's the most frequent cytotoxin found, and nitrogen influences microcystin concentration. So last week we talked about nitrogen and phosphorus, and I know a little bit, and I know that Ashley had talked about it even beforehand. But for microcystin concentration, I'm gonna go back a slide so you see there's a lot of N here, a lot of N, nitrogen. So nitrogen is needed to form that compound, right? And then you have more than 250 variants. And here's a normal liver, and here's a liver after toxins. So that just shows that the liver's destroyed, okay? Now, this is the last slide on compounds you'll see. These are the neurotoxic alkaloids. So these are the neurotoxins, right? Look at the half-life, 1 to 2 hours. Let me go back a second here. This is days to 10 weeks, right? Back again, I'm sorry, I'm going back and forth. 1 to 2 hours. And these they're a lot more toxic. And so this is where we talk about that sometimes probably these deaths that we see are probably because of these compounds, but you see how small they are. And also they degrade so quickly that that they're degraded from the water before you're able to analyze them in the lab already, okay? So just for an example, just pulling this together, again, we have anatoxin, microcystins, synaptomopsins, and saxitoxins. So you see they're produced by all these different species here. Last week, you saw pictures of these, if you guys were in here. The health effects. So you see that a lot of times the numbness, dizziness, paralysis, like the neurotoxin, again here, paralysis, nausea, vomiting, diarrhea for hepatotoxin, nausea, vomiting, diarrhea for hepatotoxin as well, for the different ones. So a lot of times if you have numbness or dizziness, or you're paralyzing or vomiting a little bit, if you go to a physician, they will not first think it is a cyanobacterial compound. So I think there's a lot more public health effects that are happening, which people are not reporting because many physicians aren't educated in cyanobacterial toxins. What I want you guys to see here is that I showed you a couple compounds, but there's a huge list of compounds here. You know, if you want to look at Paul Zimba's work, what I found interesting is that one called Jamaica Aid. So, people just name these things as they want to. There's a Jamaica Aid, there's a grassy statin, grassy peptide. But here, just to see that, that they affect blood, brain, embryos, a lot of different things they affect. So there's a huge variety of compounds. And you hear a lot about microcystin, and I talked about microcystin, anatoxin, saxitoxin, but there's a lot more out there that are potentially harmful. How are we exposed? Okay, so it's a question of how we exposed. Well, via drinking water, okay? Aerosolization. Vertical transmission. So in Nile crocodiles, the microcystin has been vertically transferred. Again, this is probably aerosolization. Recreational exposure, liver failure in dialysis center. So these are different ways that we're exposed. Again, here is to show that I told you guys that it's not new. So if you look at these two graphs here, you're seeing animals, non-human, and humans affected. Okay, so you're looking at several different animals have been affected from 1900 to 2020, and you see an increase of animals and an increase of humans being affected. And this could also be because we're identifying what it is. This is Susie Wood's work from 2016. And to finish my part, I've got 3 more slides. So now you understood public health, but we have another problem. We also have crops, and we also eat the crops. The future is blue-green, and there is an increase of toxic cyanobacterial blooms in surface irrigation, which can cause crop damage. These toxins also affect physiology of plants, and there's also potential accumulation in leaves, fruits, and vegetables, okay? So here, what I just want you guys to see is there's been work done on looking at lettuce, arugula, cabbage, broccoli, rice, and they do notice there is some increase in the leaves or in the roots. And you guys can contact me and I can send you this, but I just want you guys to see that there is an increase. There is a sometimes maintain these, and this Mickey LR is microcystin in these fruits or vegetables or leaves. Here's another study looking at microcystin dermal spray. So they sprayed the leaves with microcystin, and you see that it does affect the leaf structure, right? And here is on shoots of Medicago of the sprout. And if you look at control, microcystin LR, LW, these are those different variants, antitoxin, and crude. Crude is the pure bloom. You're seeing that there is a problem. There is a decrease. There's malformations. And if you look at the graph, the length of the root, there's a decrease with these different compounds. So there's a significant decrease in root length and seedling growth. Okay, so we have effect on health and we have effect on crops. So before Vincent takes over, again, in Florida, last week I showed you guys this, that there's several genera that are found, and these genera produce the different compounds that, these different cyanobacterial compounds, different cyanobacterial toxins, which I just showed you about, okay? So now, Vincent, to you.
All right, thanks. Thank you, Dale, and I'll get started here. Let's see. How's that look? You see my, okay, great, thank you. Hi, my name is Vincent Encomio. I am a Sea Grant Extension Agent for Martin and St. Lucie counties down here on the Treasure Coast. And Dale's talk was a perfect segue to some of the effects of cyanobacteria. Particularly what I'm interested in and presenting here is the effects on our pets, specifically dogs. All right, whoops, there we go. Okay, so just before we begin, I just want to qualify my expertise. I'm not a phycologist like Dale, nor am I a veterinarian. And I have not stayed at a Holiday Inn. So, but I owe a lot of thanks to the information I'm going to present to you today, to the people that are shown here in the slide. These are actually the local veterinarians that worked on many of the dogs that were afflicted with cyanobacterial poisonings in 2018, specifically the names in yellow. And I learned a lot from them, and they helped me produce some educational material that I hope that will be useful not only here locally, but throughout the state of Florida. So where I am is, if you're not from the Treasure Coast, generally Martin, St. Lucie, Indian River, and Okeechobee counties, I am here in Martin County, and we have an estuary, the St. Lucie estuary, shown in the two boxes in red. It is artificially connected to the Lake Okeechobee through these two structures. The S stands for structure. There's a 308 structure and the S80. We usually call the S80 structure the locks. And those are connected via a canal called the C44 canal. Every wet season, we worry about whether or not the lake levels are going to get too high. And if they are too high, they release water to the east, to the St. Lucie River, and also to the west in the Caloosahatchee River. And the picture on your lower right shows what some of those discharged lake waters can look like when it's particularly bad. And if you look to the far right, past that spit of land, that's actually the ocean. So this is water running out of the St. Lucie Inlet. So, and then when we get all that excess of fresh water, we can get things like this. So these cyanobacterial blooms, those two photos were actually from 2013. And when we get events like that, and if it's particularly bad, you'll see these signs posted at particular points along access points along the St. Lucie River. And these are put up by the health department. and they're regularly testing for bacteria, for Enterococcus bacteria, but they also keep an eye on blue-green algae or cyanobacteria blooms as well. And as Dale was saying, these are, I'm going to focus more on microcystins because it's a little bit simpler for me, but that was the main focus of what I'm going to talk about today. And the World Health Organization has 2 advisories, one for recreational contact at 10 micrograms per liter or parts per billion, and for drinking water, 1 microgram per liter. EPA, the latest regulations that I have from 2015 is a little bit more nuanced, but it's still around that low 1 microgram per liter acceptable levels. Beyond, when you get above that, it exceeds those health advisories for drinking. And that's important for dogs, and I'll get into that in just a second. These are some of the health effects that are shown with microcystin and then a couple other cyanobacterial toxins. Just know, Dale just showed a little bit of that. Just know that these are similar health effects in dogs, particularly liver failure. And the 2 main effects that we saw in 2018 with the dogs that got poisoned was acute liver failure and internal bleeding. Outwardly, and that was after examination, but outwardly signs, again, as Dale mentioned, these are somewhat generic signs. And so unless doctors or veterinarians are attuned to these events, they may not automatically diagnose these dogs for cyanobacterial poisoning. So it's very important that the public health and education components of this issue are front and center so that we can capture these kinds of events. So going into 2018, the cyanobacterial blooms started in Lake Okeechobee, kind of in June and summer 2018. Just red means very high cell concentrations. This is a aerial image just to give you an idea what that looks like, that you can actually see that in these the green in the aerial images. We started seeing Lake Okeechobee releases to the St. Lucie estuary, and really right at June 1st is when they started. And the black line shows the amount of water in cubic feet per second that was released from the lake through that C-44 canal that I showed you earlier, and eventually into the St. Lucie estuary. Like just so you have a sense of what a cubic feet per second means, in terms of the amount of gallons per day, 1 cubic foot per second is over 600,000 gallons. So it can be a lot of water. As the DEP tracked these blooms, you start to see reports from DEP, from the Florida Department of Environmental Protection. In early June, you start to see blooms around the St. Lucie locks where that S80 structure is, that eventually opens up in the St. Lucie River. By July, there are a number of dense blooms found throughout the St. Lucie, particularly if you look at that bottom photo. That's a canal community where the Florida Sportsman magazine office was. So that was well documented. And then August, blooms are still continuing. By September, those blooms are decreasing. All this information is on DEP's blue-green algae dashboard or website. So, and just some highlights of microcystin levels. These are some, we were seeing some very excessive levels of microcystin as measured by DEP. And just keep in mind those World Health Organization advisory levels for recreational contact. The recreational contact is probably the important thing here because we're not drinking the water out of the St. Lucie, but dogs swim in it and they ingest a lot of water when they do. So those are some exceptionally high values. In 2018, we had 6 cases of dogs where it was verified that they were poisoned by cyanobacteria. With acute liver failure and internal bleeding, all had noted contact with the St. Lucie River. They likely drank their water, and if any of you have dogs, you know dogs lick their fur. There was one instance where a dog chewed on a dead catfish. Vomiting occurred within about 6 hours, a half day of exposure. But interestingly enough, and this really relates to what Dale said earlier, in most of those cases, little to no visible signs of algae were present. If you recall what Dale had said with cells being lysed, but toxin was high, microcystin has a very long half-life. And I'll talk about that right here. The great thing is that all those veterinarians that I showed those names of earlier, their aggressive treatment saved 5 of those dogs. 1 dog unfortunately died, but the owners were willing to put up the expense to do a full autopsy, and they got a lot of information from that. One of the things that came out of these veterinarians' work is that they realized they could diagnose vomit and urine for both cyanobacteria cells in the vomit and actual microcystin toxin in the urine. And this is the first time that urine was used as a diagnostic for microcystin. Just to give you, again, very high levels, over 30 micrograms per liter of microcystin was found in the urine of 1 surviving dog. And speaking to that long half-life, it was detectable in those living dogs in their urine about almost 2 months later. It was confirmed that these dogs were all poisoned with Cyanobacteria microcystin, and the cost of these treatments was quite high for your average pet owner. Average cost was $3,000. We had one pet owner that had 2 dogs that were poisoned, spending nearly $13,000 to save their dogs. But thankfully, they did save them. This is some headlines. The 2018 event got a lot of attention, certainly locally and probably nationally as well. The next year, I started to see more of these kinds of incidents in the general news media. So that's even though those are unfortunate events, it is a good thing that we are more attuned to that happening. And you might see signs like this if you any freshwater bodies, like if you in a park, hopefully they're getting regularly monitored. This is a typical sign you might see in Martin County if there is a blue-green algae bloom. And the main thing that we stress to protect your pets is if you are in doubt, if it doesn't look right, when in doubt, stay out. And to go along with that educational message, been working with consultation and comments from those local veterinarians, is developing a fact sheet on cyanobacterial poisoning on to how to keep your dog safe from blue-green algae in Florida. And we getting close to distributing this fairly widely, certainly in my area and hopefully throughout the state. So if you're interested in getting a copy of this, just let me know. But the last thing I want to end with is just a little PSA. Just remember that to pick up after your dog, because we can't train our dogs to do this. 1 gram of dog poop has over 23 million bacteria, and a little over 40% is found of dog owners do not pick up after their pets. And what that might mean in our area, which is generally overall the Indian River Lagoon, that could mean over 100 tons of poop per day that might not be scooped. And even though not all of that washes into our waterway, we want to do the right thing. We don't want to be the person that eventually might pollute a waterway that could lead to a blue-green algae bloom, and then eventually our dog getting sick. So, we all want to do the right thing and make sure that we don't become part of the problem, because that excess poop is what helps fuel, with all the nutrients in there, what helps fuel can help fuel those blooms. So with that, these are some of the resources I've put together. Some of them are on that fact sheet that helped inform it. And I think with that, I'm good to go. If you guys can start, I think Laura's going to put together a quick poll.
Yeah, you should be able to see the poll on your screen right now that just indicates whether you learned something from today's webinar and how many people were watching on the device with you. It helps us track our progress. You'll also be receiving a recording of this webinar or an email with a link to the recording of this webinar so that you can share with other people who might be interested, which also includes a link to an additional evaluation that we appreciate getting that type of feedback too. It helps us know what topics are popular with bite-sized science listeners. So I'm going to go ahead and end the poll. Vincent, if you want to proceed to your last slide.
Oops, let's see. There we go. Okay, so yeah, if anybody has any questions for Dale and myself.
I'm not seeing any questions in the chat right now. I thought both presentations were excellent. It is 4:30, 3:30 Central Time. We recognize some of you may have to go, but if you do have a question, please, we'll give it a minute here to chat, put it in the chat box. And Dale and Vincent have agreed to stay and answer any questions. So far we've got a thank you so much. And I know.
Thank you. You're welcome.
Welcome. Thank you.
So how often do they, how many times a year does the Lake Okeechobee waters get released into the St. Lucie?
It doesn't happen every year, but the threat is there. If we get, one thing that I've noticed over the years since I've lived in Florida is when we can get these early rainy season high rainfalls like we had this year. What's saving us right now is the lake levels were very low prior to that. In fact, it was not a good thing. Actually, there was widespread drought throughout, quite a number of areas in South Florida. But what was significant about 2018 is that was the third consecutive year of damaging freshwater discharges. And then 2018 was also really significant in that both the East Coast and the West Coast had cyanobacterial blooms. The West Coast actually had it the worst in 2018 because they also had red tide in the Gulf of Mexico. So that was I hate to say, a watershed year for harmful algal blooms for South Florida. So we dodged a bullet in 2019. We did not have any significant cyanobacterial blooms. So it's, there are specific years. One of the things that's an important factor is the El Nino La Nina cycle. So some of the really damaging ones, the 2 years that really stand out in our area for Lake Okeechobee discharges is 1998 and 2016. Both of those were very strong El Nino years. And you get a high amount of rainfall during what's supposed to be your dry season. And so that sets the stage for the ensuing rainy season.
So we do have a question in the chat box. How far away from the actual release water can microcystis travel and be harmful?
As an example, in 2016, the blooms were so bad that they got out the inlet, the St. Lucie Inlet, into the ocean and actually caused beach closures throughout Martin County because it was heavy enough. I don't know what the microcystin levels were at that time, but visually, and that's really the standards that are used to inform these health advisories is visual. They were heavy enough that they closed beaches throughout all the way, maybe even a few north of the Martin County line into St. Lucie County, but certainly Martin County beaches were closed.
Yeah, the kinetics of microcystin in marine saltwaters I think they're starting to study some, but it's not very much studied because historically you weren't looking at microcystin in marine systems, right? Or saltwaters. There's some work done by Rafe Kudela in the California coast that does a lot of that work. And I just saw that you may follow up, Laura, with the question, can it be harmful if you can't see it?
Yeah, go ahead.
Okay, so Vincent made a comment, of course, looking at the way Florida does it is looking at, okay, if it's green, don't go in there, right? That's the Florida way of the way that they the way that Florida does things. And if someone's here from other states actually have numerical values that they go out before they close or they open or whatnot. In Florida is more of a, it's more of a visual, exactly what Vincent said. And maybe Vincent, you can come back on me on that. And then, but you're correct. So like we talked about, and Vincent made a comment as well, the toxin, the visual coloration of the blue-green scum or that color can go away. However, the compound, if you're talking about microcystin, it is still in the water, right? And again, we have 250 some variants, and the stability can be different. And again, it depends on the water quality, depends on how the UV radiation, depends on how much ozone or peroxide is within the system, and all that stuff, like natural in the water. Yeah. So I guess my long answer to your direct question. Yes. So the water, if you're considering harmful as toxin having microcystin presence or microcystin concentration, it is actually more harmful for microcystin after the bloom has gone away. Does that make sense? Right afterwards, because you lyse the cell, the toxin comes out, and then it's even more harmful. Again, but okay, going back, depending if you're looking at recreational exposure or water or for water intake, because recreational guidelines are on total microcystin. That means, dissolved and intercellular. I mean, both in and out of the cell, right? So again, and this is where you go back to water work. So does that make sense? Oh, yes.
I think it does. And it's also another good reason for the education campaign that we're working on. Because like you said, these veterinarians or even human healthcare providers may be seeing people with symptoms, and they haven't seen, nobody, thought to think about the color of the water, and they don't know to even test for it.
Correct. And then, like I said, and then going back, Betty, to your question, it's important when you do like management techniques, you're looking at, okay, what is the water used for, right? Is it for drinking water or is it recreation? Because then you go back to back to what Vincent showed about the numerical. Is it total microcystin? Is it intracellular? Is it extracellular? Because then that depends on, that becomes a larger question afterwards. But for total microcystin level, correct. It can be harmful. You don't see it.
Well, very good. I really appreciate both of our fantastic speakers today, and appreciate our listening audience as well. So I think we'll wrap up this series of Bite-Sized Science. Be sure to go to our website to check out our upcoming webinars on Tuesdays and Thursdays through the month of July. Everybody have a great day.
All right. Thank you.
Thank you, guys.